Bismillah ar-Rahman ar-Rahim. This is the cartilage data show. First of all, you should know that the cartilage is a type of connective tissue. The cells of the cartilage originate from the undifferentiated mesenchymal cells, like any connective tissue cell. Also, it resembles the connective tissue in the fact that the cells here are widely separated from each other. Here we have the cell and the other cell, and these cells are widely separated from each other by the matrix. The matrix here is characterized by being rubbery. Here, very important feature is the rubbery consistency of this matrix, which makes it firm but flexible, and this is different from the hard, inflexible matrix of the bone. Very important character, it is non-vascular. Also, it has no lymphatic vessels, and it has no nerves. So, as a summary, it is a certain type or specific type of connective tissue formed of cells and fibers and matrix. The cells originating from undifferentiated mesenchymal cells. The matrix is rubbery in consistency and this makes it flexible, and it is non-vascular, also devoid of lymph vessels and nerves. Cartilage is classified into three types: hyaline cartilage, yellow elastic cartilage, and white fibrocartilage. And this is according to two factors. First, amount of matrix between the cells. Second, the type of the fibers embedded in this matrix. This is the high power for the hyaline cartilage, which is the most common type. It is called hyaline because when it is in the fresh state, unstained, it appears transparent like the glass. Hyalo in Latin means glass, so it is called hyaline cartilage because in the fresh unstained form or state, it is translucent like a glass. Here we have the structure of the hyaline cartilage. Here we have, this is the perichondrium, which is the outer covering of the cartilage. This is the peri means around, chondrium means cartilage. So this is the outer protective layer for the cartilage. This layer here, it is formed of two elements. We have here outer fibrous layer. This is the outer fibrous layer. Here it is formed of the collagen fibers, fibroblast, and this layer is characterized by two things. The type of the collagen here, it is of the type 1. It is of the type 1 collagen. Very important to know that. It is collagen type 1. After that, you should know this is the collagen type 1. After that, this layer, which is the fibrous layer, is rich in blood vessels. It is rich in blood vessels, and this is very important for the nourishment of the cartilage because the cartilage is avascular. So this perichondrium supply the cartilage with the nourishment that will pass to the cartilage by diffusion. The second part here of the perichondrium is this layer of cells. These cells are actually chondroblast. Blast means it is an active cell that will produce cartilage. Chondrocyte, chondroblast. So this is the inner chondrogenic layer. Genic means form. This layer is formed of the chondroblast, these cells that later on, when there is a need for the growth or repair of cartilage, these cells will form the cartilage, new cartilage by formation of matrix fibers and being mature to form the chondrocytes. After that, here you may be asked in this situation about the functions of the perichondrium. Here you should know that the functions here is divided into two. First one is the nourishment or nutrition of the cartilage, which is the function of the outer fibrous layer. Then the second function is the formation of new cartilage during the growth or repair. So this is the function of the inner chondrogenic layer formed by chondroblast. And the third function, it is the site for the attachment of the muscle. So these functions should be remembered because it may come as a question in the exam. After that, here in the picture, come again to the picture, here we have the cells in the periphery of the cartilage. These cells at the periphery of the cartilage are called chondroblast. So we have chondroblast in the inner chondrogenic layer, and also we have chondroblast, which are immature young chondrocytes, present also at the periphery of the cartilage. So these are the chondroblast at the periphery of the cartilage. In addition, we have chondroblast also in the perichondrium, in the inner chondrogenic layer. In each cell here, you should know the origin, the site, LM, EM, and functions. So, as regards the origin, it's very known that it is one of the cartilage cells, so it originates from the UMC. Then, as regards the site, it is present always at the surface of the cartilage, as well as the inner aspect of the perichondrium. As regards the LM, it is oval in shape or spindle in shape. This is the shape. And it has a deeply basophilic cytoplasm with a large oval pale nucleus. And as it is an active cell, this large oval pale nucleus has a prominent nucleolus inside because it is an euchromatic nucleus. This cell can divide, so the nucleus very pale, very euchromatic with prominent nucleolus. As regards the EM, you can expect that this cell should be rich in the protein, the organelles responsible for protein synthesis. So we can expect it has a well-developed rough ER, Golgi apparatus, and mitochondria. So these are very fixed features for the protein synthesizing cell. In addition to that, it has also abundant ribosomes for making the proteins for itself. It is a dividing cell. Also, being an active cell, it should has the euchromatic nucleus. So this is as regards the EM picture. As regards the function, of course, it forms the cartilage, so it is responsible for the formation of the matrix. First of all, it forms the matrix. Formation of the matrix itself, and once it is surrounded by this huge amount of matrix, it is imprisoned and became or changed into chondrocytes. So it forms the matrix and it changes to the chondrocytes, the mature form. More than that, it forms the collagen present in the matrix, which is of type 2. Take care. The cartilage fibers, collagen fibers in the cartilage in the perichondrium is of type 1, while the type of collagen in the collagen fibers in the matrix is of type 2. After that, it has the function of the growth from the surface, which is called the appositional